So the next speaker will be Barenholz. I think I don't need to introduce him, and he will introduce us into lessons learned from Doxil. So thank you very much for coming.
Oh, my pleasure. Thank you very much, Jan. As usual, it's really a great pleasure to be here. I think it's really the mixture of people here make this conference unique in a way. It's small enough that you can enjoy everyone, but big enough that you can learn a lot of things. So I think it's really fascinating conference. And it's also the opportunity to thank very much Patrick and Biet for the wonderful job they are doing with this conference. So thank you very much. When we work on Doxil, when started working on Doxil, which was 1987, long time ago, after our failure with the first formulation in the clinics of the liposomal doxorubicin, actually I wrote a notebook. And in this notebook, I decided that everything which is not important for the development, I'll write down and try to solve it later. And I did it. And I think a few months ago, 2 months ago, I finished the last point. I still don't have all the answers, but at least I believe I know most of the questions, and that I think is very important. So actually, what I want to share with you is one of these last points that we are trying to deal with or dealt with, and this is the two aspects. One is the effect of the microenvironment of the tumor on the drug delivery system. How can you utilize it? Can you improve? Can you modify, control to some extent the microenvironment? That's question number one. And question number two is, what is the optimal release rate of the drug? Okay, these are the two major questions we are trying to answer. You know, I think I showed these slides many times. And basically, our main point of using drug delivery system is to get us to the point that we can get more drug into the tumor and less into irrelevant system, tissues. But we know today that that by itself is not enough. Actually, we know it for a long time. We need the drug to be released. And the question is, well, okay, released, but how much, to what level, and how. All these are questions that are still not resolved in a good way. So to do this, we have to find a way to get a better release or different release in the tumor site. Okay? Again, I want to stress the point that when we are developing the system like that, what we want to achieve is a liposome or any delivery system that will circulate, loaded with a drug as long as it's needed to, and then when it extravasates, it will extravasate to the tumor site with the drug in it. That's critical. But that can be a dead end, as it was with the cisplatin formulation that Alza used. And when this was done, they got excellent tumor localization in patients, no toxicity at all, but zero efficacy as well. And the zero efficacy resulted for one major reason, that there was no release at the tumor site, or release was minimal. So we know that when there is no release and there is no uptake of the liposome by the tumors, which is in most of the cases what's happening, you will not have any efficacy. So the question is, okay, how you get now this last parameter to work, how to get the system to do. I don't think I have to convince you why we selected liposomes, and if I'll do the work today, I'll still go for liposome, for the very simple reason that we know a lot. We have a lot of information about the system, and I think it is for the young people now to bring all these systems that I put here on the wall, on the board from this paper, and many more, to bring them to the level of knowledge we have with liposomes, and then I think they will be as attractive. I think that's a lot of work, but I think it's a work that should be done because I'm sure that some of these systems are very promising, maybe even more promising than the liposome. But this is the reason that I selected liposome. I don't want to go over it in details, but basically a system that we know a lot about it, and because of it, it's easy to work. You can just go and start doing your design work and of the formulation and then do the biological work. So let's keep it. I talked about it. We all know that, and I think I try to stress it in any conference, that tumor has one aspect that many people tend to ignore, especially many physicians. That's and Patrick, excuse me, that's the physics of tumor that really work against us, unfortunately. The tumor has high pressure inside it. This pressure reduces the regular diffusion of drugs from the blood vessels at the tumor. So the concentration of the drug in the tumor is actually lower when you speak with low molecular drugs, lower than it is in other tissue. And because of this, you lose. You have to give more, then you raise toxicity and so on. So I think the problem is how to overcome it, and that's what liposomes we found they are doing. This is the EPR effect. I don't want to go into details, but just remember it has two sides to it. One is the porous blood vessels, the new blood vessels in the tumor site, in the tumor tissue, and the other is the lack or the reduced lymphatic drainage. Both, again, work in our side. So to design a system, all this you know what we have to achieve. I just added one point this year. And this is, we need to go and understand better what is the optimal release rate we need. And then the second question that I asked, that I said before, how the microenvironment is involved with all our aspects. So actually, Doxil show all the above three parameters, remote loading, stable remote loading that enable to bring the drug to the tumor. Now we know that actually we get release of the tumor. Actually, we know it for a long time. We achieved this with the pegylated liposomes, and I have to say here the opportunity is that I'm glad that Terry Allen is with us because she's the really pioneer of all the concept of stealth, and she's the one that started all this, and she deserves a lot of credit for it. The extravasation, which related to the small size of the liposome that enable to get the EPR effect, and what I'm going to talk more about is actually the last point. So actually, we know that we're dealing with passive targeting, but active targeting we know will have the same barrier actually that passive targeting, and we have to overcome the same obstacle. And I'm going to go now to the story of 12. So this was the objective, to study the two parts. We use our classical dogma. I always show it. Look for performance first. What do you want to achieve? What are exactly you want to achieve? Based on this, you decide on what property you need your delivery system, on the structure and the processing. Not always you can get all what you want, but at least you have to try. And in order to do it, you use this kind of concept map that has the design and science part on the left side, the basic science, and then the biology part on the right side, where you try to improve your therapeutic index. And I'm not going to go into detail now to the system. Actually, what helped us a lot is that we didn't have to look for a system, because we have one system that was designed by Dave Needham many years ago. I hope that Dave is here. And I know he's in the conference, and we have Doxil. We know that we can achieve a very fast release with the system that David invented and actually introduced even into the clinics with the help of Celsion. And this is the system that composed of DPPC, monosteroyl PC, and PEG DFPE. So it's a pegylated liposome, and it's thermosensitive because it undergoes phase transition. It has a phase transition at about 42 degrees. a little bit less, and this is the phase transition. So upon exposure to heat at this temperature, there will be a very fast release of the drug, and if you do it at the tumor site, the drug will be released there, and then you know what happened upon fast release. And this is the classical Doxil. It is the liquid order phase. There is very little effect of temperature on the release, a little bit, but much less. You don't have a burst release type. And this has cholesterol in it and HSPC, no DPPC. And the cholesterol is what's responsible for really keeping it very intact and with very minimal leakage of drug in the plasma. So you can see we prepared this liposome. We loaded by the same method that proposed by David Needham, which is here we did the ammonium gradient, and here we did the pH gradient using a citrate buffer inside the liposome. So we basically copy his system, and we can do it very well. Just to show you what I said before, that we are in the LO phase, and while this is with the liposomal doxorubicin, with the Doxil, and this is with the thermosensitive formulation. You can see that at about 42 we'll undergo a phase transition that lead to a drug. I hate these student type things, really. So these are the comparison. You know, if we take the Doxil and run it in a calorimeter, in a DSC, there is no phase transition at all. But if we take the lipid, the HSPC and PEG DSP, we can see a very distinct phase transition at about 52.5, 53 degrees with the TM. Here you can see the TM in this case. And this is what happened with the thermosensitive liposome. You see the phase transition at about 42 degrees, as I said. And you can see the delta H and all this. I don't want to discuss it. So it's a very distinct phase transition. I have to. These are the two methods of loading we are discussing. This is the ammonium sulfate. It's here. And the pH gradient with citric acid is here. And it worked perfect. We got very good loading with both methods. I want just to stress that the ammonium sulfate gave us a very, very high stability. As I'll show you later, the thermosensitive liposome, because it's like cholesterol, are much less stable with relation to leakage. But this shouldn't affect our fast release, burst release upon the temperature. Here you can see the comparison of the two. And you can see in both the crystal, this is a crystal, they are a little bit different. This is the crystal of doxorubicin citrate, and this is doxorubicin sulfate. And I think you can see them both. And I just want you to remember this electron micrograph. Let's see what happened to it later. With Doxil, if you expose it to temperature, even at 50, 60 degrees, not much is happening to the shape. So I even didn't waste the slide to show it, but you can see here what happened to the thermosensitive liposome when you hit it at 42 degrees for 5 minutes. Look at the liposome, they don't contain the drug anymore. We can measure the release, of course, but this just shows it very nicely. But to our surprise, with time of being in the refrigerator, the crystal becomes smaller and smaller and then disappear altogether after a very long time. So it doesn't retain the drug very well, at least not in our hands. And again, it's not much to say about it, just to show you that even with cryo-TEM, you can follow what happened to the fate of the drug very nicely inside the liposome. So I don't have to repeat the story of Doxil, so I go forward. So this is, again, what are the parameters that are important. I just want to add one interesting parameter, and this is a nano conference, so let's look at the nano value, at what we mean by nano. And you know, now we know how to calculate very well the volume of the liposome. And we have developed a software that can do it on many, many large particles. I don't have time to discuss it, but we can see here that the volume of one liposome is about 1 in 10 in minus 19 liters. So you can imagine how small it is. It's difficult to imagine, which means that movement of one ion is enough to affect all the properties of these liposomes. So this is the impact of nanos. Very small motion, very small changes in amount can have a tremendous effect on the system. Okay, this is why Doxil was successful, and I don't think I'll repeat it. You can see it. The excellent pharmacokinetics that show, as I said before, the very large stability in plasma. If it will be in the tumor, we'll have the same stability as it has in plasma. We will not see any activity whatsoever. The release are much too slow to have any effect. And actually, in order to improve the release, at LTI at that time, they tried to, at the 1980s, beginning of 90s, they were trying to change the formulation. Let's say by changing from HSPC to DSPC to DPPC to reduce cholesterol, everything they touch that increase the rate of release make the formulation much less efficacious. So we know we need to be a robust formulation in the plasma. This just showed that we actually get an EPR effect, and actually this is a slide that one of my students, Noam, who is here also, showed that there is less uptake of Doxil by macrophages than of the regular, and I skip it, and I want to go. Okay, this is the release of the thermosensitive liposome by exposure to temperature, and indeed you see that in 5 minutes all the drug is released. And I think David Needham did a very clever formulation by adding the LisoPC that actually improve the release rate very much. I kill the students when I'm back. This is what he did. This paper appeared recently, but David knows this for a long time, that with the addition of PEG, you get really a complete release, which you don't get if PEG doesn't present there. And I think that's a very, very important aspect. So what we did now, in order to induce the release in vivo, we used thermal ablation, okay? And the system is, this is the system we were using, and actually the results look, if you do RF alone or RF with Doxil, you can see a much better effect on the tumor. You can measure it. I want you to look at the ring that we see here. This is a ring which has a kind of inflammation-like reaction, which really modified dramatically the microenvironment. This was measured by many people. So the experiment we did is two different, they have to be very different. When we injected the thermosensitive liposomes, what we did, we followed actually the protocols of Celsion in some of the papers, that you first inject the formulation, then you wait certain amount of time, relatively short time, and then you expose the tumor. These are tumor-bearing mice. You expose the tumor to the RF, and then you start doing your measurement and follow whatever you want. When you do with Doxil, you do a little bit different. You first change the microenvironment. You cannot do this experiment with the thermosensitive because you have to expose the thermosensitive liposome to the RF, and I don't want to do few exposures, so we have to do it this way. With the case of the Doxil, we first expose the tumor to the thermal ablation, and then we inject the Doxil, and then we can follow, because Doxil is not, the RF doesn't induce any release for the Doxil, so there is nothing to do. And let's look at the pharmacokinetics first. So what we can see is very interesting. If we compare the two, we can see here that the thermosensitive liposome in the liver at 1 hour show a very high level of drug. And if you look at the Doxil, the blacks, and you look at this, then it's decay, but it's very high. So I think this was the reason why Celsion decided, I guess this is the reason, maybe David can comment on this, why Celsion decided to use the liver as the really first indication for the use of their thermosensitive liposome. But if we look at the plasma, we can see actually a very different profile. If we compare, we can see that the thermosensitive liposome are at a much lower level, the doxorubicin much lower level than it is with the Doxil. And if we look what happened in the tumor, again, we see after a very short time, we have a quite nice level of drug on tumor, but it's disappeared very fast. While with Doxil, you have a build-up that keep building actually after it, and this is with the combination of thermal ablation. Always, with both formulations, the thermal ablation improved the performance in pharmacokinetic and as well as efficacy. We can see here that if we look at total drug, which is liver, plasma, and tumor, it's not really total, but it's most of the drug. We can see that the total amount that we have in the body is much higher when we have Doxil than when we have the thermosensitive liposome, and that's because the thermosensitive liposome simply are leaking. The other thing I have to say, it's very important that after the exposure of the animal treated with the thermosensitive liposome, we still have drug in the liposome, in the plasma, even with the thermosensitive liposome. So we still could benefit from the thermal ablation effect on the tissue. This is the idea. Now, this is, I think, the last slide, and we talk about the efficacy. You can see very clearly that the best formulation are the Doxil with the RF. The rest are much better than the untreated, but this is a big, big difference, and we use two parameters. One is the tumor volume, and the other is the humane point. You know, today in the animal facility, we have every day a veterinarian will come and he will weigh our animals, and if they are below certain weight, he told us to kill them. This is the humane point. And, well, we have to live by this rule. It's become almost impossible to do animal studies anymore. But by this, you can see that the only 100% survival we have is with Doxil plus the RF, and all the rest were lower. This is the control, untreated, and these are actually the conclusions. So what we conclude from this is that, at least in our case, for extrahepatic tumor, we did not test hepatic tumor, so I cannot say much about it. In the extrahepatic tumors, the fast release, the very fast release, fast burst release, is not optimal. To say that the Doxil release rate is optimal, I don't know. So what we are doing now, and that's the last part, which I cannot discuss for patent issues yet, although I wanted very much, the patent attorney warned me not to speak much about it. We found a way that we can modify the microenvironment, and now with this we can affect the release rate, and then we can use the same Doxil, doing it at a different release rate, and maybe with this we'll have answer what is the optimal release rate. And I think I'm much over my time. That's my usual way, so I apologize for it, but just to show you how much Doxil was, how much people were involved with Doxil development, many, many teams. And this is the team that did the work with the thermosensitive liposome. It's actually a master thesis of Alex, and he was got help from Erez and Lisa, and we have a lot of input from Nachum Goldberg that really introduced her to the RF, and he's a co-author, will be a co-author on this work. And I think I want to thank you very much.
Yeah, thank you very much, Gezi, for this nice presentation. So we have allowance only for one short question. Yeah, please.
Hessie, nice talk. I'm very pleased that you're following up on the points that you're following up on. But and we've found too that Doxil needs to be released. The more slowly it's released, it seems that the better the therapeutic effects in animal models. But on the other hand, Peter, who has done a lot of work with schedule-dependent drugs as opposed to Doxil, which is schedule-independent, and he's found that a much more rapid release is more effective for schedule-dependent drugs. Do you think that that's one of the reasons that we need to pay attention to release rates for these drugs depending on their mechanism of action and whether they're schedule-dependent or schedule-independent?
Absolutely. I think absolutely. So I didn't mention it because, I focused on doxorubicin, but you are absolutely right. I think it's drug-dependent. I think many drugs will be benefit a lot if you plan it according to the mechanism of action of the drug. You're absolutely right.
Okay, I suggest then that other questions are held in the lunch break or whenever with Gezi. So thank you again.